With the first talk. First talk is by Dr. Saher Mehdi. He's the CEO and co-founder of Rewise Health. They're a company that uses systems approach to provide health insights on personalized nutrition and biological age. With the help of these parameters, they're able to hopefully predict true health age. And so over to Dr. Saher. Thank you.
Thank you for the introduction, and it's a huge responsibility to be opening a research session, that too in longevity and at Indian Institute of Sciences. So it's a big dream and an ambition really coming true today. But before I begin my talk today, I want to take a moment and ask you all, can we really optimize longevity for all? So I want you all to raise your hands and see how optimistic our audience is about the future. Can you all raise your hands? Who believes you can really optimize longevity for all? So it's great. It's only about 30% of the audience believes that. So for the pessimist there, I want you to take a moment and just imagine, can we imagine a future where living to 100 is not just a milestone for lucky few, but a norm for all? A future where aging is not a barrier, but a testament to life well lived. A life full of vitality, purpose, and health. So, as my introduction said, I want to say that I'm not just an entrepreneur. I'm trained as a molecular biologist and have been studying aging and stress mechanisms in biology for half of my life. And only recently I have moved into industry. So what I'll talk about today is how we can translate what is happening new in aging biology and convert it into usable everyday tools for everyone to optimize longevity at scale. Okay, so what if the secret to aging or secret to healthy and fruitful life wasn't stored in some fountain of youth depicted in picture, or some mystical elixir that we are out there looking for it, but actually the secret to longevity is encoded within our own biology, waiting for us to unlock it. Advances in science, precision medicine, and technology are taking us closer to a future where living to 90 and even beyond 100 is not just a possibility, but actually achievable for all. But is it just about adding years to our life or adding life to those years? The real question is, can we provide a equal opportunity irrespective of our genomic library or social economic status to all? We are at an inflection point in aging biology where our understanding and approach to aging will change. And we are no longer at a point where we are just treating aging, age-related symptoms like Alzheimer's or diseases like Alzheimer's or frailty or problems with eyesight and things like that. We are actually talking today at the brink of a revolution of actually reprogramming the very core of aging, perhaps delaying it and even reversing the cellular mechanisms that drive it. But the real question lies, with all these advances in science, how do we make sure that they reach every aspects of the society or every sections of the society? And how do we translate the cutting edge science and translate into everyday tools for human life. So today, I want to really make an attempt to shift this notion that age is an inevitable decline to a time where aging can be treated as a period of continual growth and vitality. So what is the biggest hindrance in optimizing longevity for all? Age is a universal fact. We all have to age, but we all age differently. Some of us are fast agers, where others are slow or normal agers. And this is because aging is predominantly, or 80% is an epigenetic trait and is driven by epigenetic factors. Factors that change the function of our genes, not really change the structure of our genes, but change the function of our genes. Epigenetic factors like our diet, our lifestyle, our environment, and most importantly, our habits. These are the factors which are driving the aging process. When we talk about variability within aging, we see this across the board. If we look at an interracial level, there's a variability between white population and diverse population like Indians. So a recent study showed that, published in Clinical Epigenetics, showed that there is 76% differences in the DNA methylation patterns between South Asians and Europeans living in London. And that's huge, 78% changes in DNA methylation. And if you just imagine, all these aging clocks we have talked about today are dependent on this one measure, DNA methylation. And it's so different between Indians and Caucasian populations. There are differences within genders. There are inter-individual differences. Male and females are fundamentally built very different. And that's because of the contribution of the X chromosome, also because of the contribution of the hormones and disease risks. At organ level, we know all our body organs do not age at same rate. And further, this complexity gets even deeper when we talk about each cell. Each cell is aging at its own speed, has underlining that, and there is variations in molecular mechanisms, whether it's connected to DNA repair mechanism, telomere iterations, or mitochondrial differences. Every cell is taking its own path. And the point I'm trying to make today is a lot of these aging clocks that are out there are either oversimplifying it or generalizing the output that we are getting, the biological age calculation then we are getting. And they are often designed on a very specific data set that is Western, male, and of a specific group. So whenever we are talking about aging as a trait to measure, we have to keep the context of variability in check. So how do we understand this complexity or address this complexity and bias? One of the major things we have to begin is we have to be very careful in picking what aging clocks we use. And we have to choose the aging clocks based on the context or the utility. Not all clocks were designed to be same. Some aging clocks were designed to predict mortality, which might not be very good in preventive health setup. So here is a beautiful graph by McKinsey Health Initiative that kind of summarizes what can be done to tackle this problem of faster aging. As it shows, we need to start by elevating or lifting the quality of life. And when I say that, we need to measure the rate of aging very early on, and only then we can enhance the time spent in good health and then in turn affect the health span. So at Rewise Health, we understand this ambition of optimizing longevity for all is an ambitious task that would require scientific rigor, meticulous planning, and help of technology. So we are basically developing a longevity diagnostic platform, which we think is a breakthrough in understanding longevity. It's an AI-based, longitudinally integrated platform, which brings various types of health data and then kind of de-identifies them, and then we apply our proprietary algorithmic method to generate longevity insights in form of biological age calculation and system age calculations and various things, so on. Once we have the longevity diagnostic scores, we use this to drive interventions, and that is also driven through an internal intervention engine that we have developed at Rewise Health. And the point I'm trying to make here is that this platform is really based on the context that we choose. So if it's for a healthcare provider, we provide longevity diagnostic based on blood or clinical markers of health. If it's for an insurer, it would be different. And if it's for a transplant specialist, it would be different. Okay, so how do we translate this technology into an approach for everyone? We take a systems biology approach, or systems biology bottoms-up approach, whereby we measure aging, and then we correlate these insights of aging to what are the pro and anti-aging markers, and then we can thereby optimize health. And last and not the least, with this technology and approach, we did an initial pilot study of 220 people with some cardiometabolic risk, and found that within a 6 months period of longitudinal study, we can reduce biological age by 4 years. We also found interesting differences in males and females. For example, in males, the key factor driving faster aging was mitochondrial dysfunction, whereas in females, it was dysregulated nutrient sensing, inflammation, and gut dysbiosis. So these are very interesting points that we need to keep in context when talking about aging. So just closing my thoughts today, I want to say that the future of optimized longevity is in community-driven wellness. And this is well evidented by the blue zones that we all are aware of. So it doesn't really matter, as Harvard, 85-year-long Harvard study says, apart from preventive care, the key driver of longevity is the quality of our relationships and what we do in those extra years of life. Thank you all for your time and this opportunity, and I'm happy to take any questions.
Thank you, Saher. We have time for 2-3 quick questions. There's one in the back there. Please go ahead. Please introduce yourself and just ask the question.
Hello, good morning. Wonderful talk. My name is Sumeru. I'm a PhD student here. So when you talk about biological age, and when you talk about it as a systemic thing, saying you could reduce it by 4 years, is there a way for you to quantify age based on organ systems or connective tissue? And when you say 4 years of reduction, does it apply to all of these organ systems, or are you using some markers in the blood to say that their age is reduced?
It's a very good question, and I think the answer just lies in the fact that when we say biological age, aging itself is a systemic process. But as I said, there is variability within the body. Each cell is aging at a different speed. Each organ is aging at a different speed. So when we talk about biological age as a parameter or a metric to measure your health, we are talking about overall average of whatever your whole body system. So it's an average estimation. It will never be as accurate that your liver is at 56, whereas your heart is at 70 or 80. It would never be that perfect, but it is always an average. So we have to always understand we are talking about a systemic marker, then it will always be a global average of your body parts.
Thank you. We have one more question here. Go ahead. You third. Go ahead. Ma'am?
Just wanted to know, you are monitoring, how long are you monitoring the person for? Because you're saying you're reducing the age by 4 years, and what are the interventions? Are those therapeutic interventions, or those are just physiological interventions that you're doing?
So I think you asked two questions here. About the pilot, I'll say it was a 6-month long pilot. And I think that was not a long enough period, but still, because we measured the biomarkers at the start of the study and at the end of the study, we could see the changes, the visible changes in biological age by 4 years on an average. At a SaaS platform level, we are actually providing the longevity diagnostics and recommendations, and the SaaS platform is designed to be integrated in preventive healthcare for hospitals, for insurers, for wellness clinic, anybody who wants to really understand and need those diagnostic tools, we provide them that.
Last quick question.
Yeah.
Yeah.
So I'd like to understand your clock a little better.
Okay.
Does it involve only metabolic parameters, or have you also integrated some genetic markers and some microbiome markers into a personal clock that you've tracked for these 220 people?
Okay, so an excellent question, because I didn't have time, so I had to rush through. I had a much bigger plan for my technology section, but I was running out of time. So the specialization of our diagnostic platform is that it is data agnostic. When I mean data agnostic, you can pick and choose what you want to calculate in the end. For example, and I think that's where the context of utility and context of variability comes into play. For example, at a healthcare provider level or at a preventive care level, they don't want to know your DNA methylation pattern because they can't do anything with your DNA methylation pattern. We still do not know what should we eat to decrease our DNA methylation or increase our DNA methylation. For them, it's the blood markers, it's the clinical markers that use. So the platform has the beauty that you can just bring in those 80 blood markers. We handpick the blood markers that we need for our algorithmic calculation and then generate a biological score for the clinician to use. Similarly, for a transplant specialist, when we get tissue specific, then we measure transcriptomic data. And this can be a multimodal data. So in future, our ambition is to basically have a luxury kind of a longevity clinic where we can measure all these markers and create a gold standard of age metrics, so to say. And that would have a multimodal collection of both genetics and epigenetic markers of aging.
Thank you.
Thank you.
We'll stop there. Sorry.